Hello and welcome to this research proposal where I'm going to be talking about skeletochronology and endocrinology to find age and maturity of sea turtles. So endocrinology is going to be finding the maturity and skeletochronology is going to be finding the age. So what do we actually already know about the age of sexual maturity? Well, we know that it ranges based on the different populations that we look at. So if we're looking at leatherback turtles, they're estimated to be around 9 to 14 years old. Hawksbill are around 17 to 24 years old. Olive ridley are around 14. Kemp ridley's at 13. Loggerheads, talking about the two different populations, Mediterranean population of loggerheads is 23 to 29, but the Atlantic population is 25 to 35. Flatbacks have very little research done on them, and it's seen from the years of 7 to 50 they could be sexually mature. But for our Hawaiian green turtles here, they're around 25 years old whenever they are sexually mature. So what does sea turtles mean to the environment? Sea turtles are the lawnmowers of the ocean, as Cameron Allen always says. They graze on seagrass, which stops the buildup of nitrogen, which allows for the transport of nutrients throughout the environment. So how is sex actually determined in sea turtles? Well, obviously, we would look at genetics. We'd look at the sex chromosomes. Well, actually, you can't do that in sea turtles. Humans have sex chromosomes, but sea turtles actually do not. Their sex is actually determined by the temperature. So TSD, temperature sex determination, is actually where, based on the air temperature when these turtles are being incubated in the sand, that'll actually determine whether or not the turtle is going to be female or male. When the temperature is higher, it's going to be more female, and when it's lower, it's going to be more male-based. And there's no external clues until the turtle is actually sexually mature. These photos here, on the left, we have a male turtle as it has a really long tail, and then on the right, we have a female turtle because it has a short tail. There are two processes for endocrinology in sea turtles. We have RIA, which is radioimmunoassay, and we have enzyme-linked immunosorbent assay, which is ELISA. RIA has been used since the 1970s, but there has been a switch in the mid-2000s to the ELISA method because it is seen to be safer as it doesn't use any radioactive material that the RIA method does use. For both of these processes, the first thing to do is to extract and isolate the hormones from the plasma itself, and this allows us to not have any other contaminants or hormones that we don't want to analyze actually attaching to the antibodies, so we get an inaccurate reading of the concentration itself. On the right, you can see concentrations being quantified for the testosterone, and their scales both on the left and the right side from low concentration to high concentration. Higher concentrations of testosterone, around the 2000 picograms per mil, you can see are more clear, but if you look at the 0 picograms per mil, it is more of a yellow color. So between 100 and 300 picograms, that sex of the turtle itself could be male or female, but above 300, it is most likely going to be a male, and below 100 it's going to be a female. This is not just an eyeball test, this is just an example to show you guys. These plasma antibody solutions are actually run under machines and actually given us the concentrations that way. Skeletochronology allows us to find the age of an organism by analyzing the lines of arrested growth, or the lags. The first experiment was done by Schroeder and Baskin in 1968 on bullfrogs. This process is very similar to looking at tree rings and counting the age of a tree. So this picture on the right here is an ossicle from a leatherback from Avens et al. Study in 2009, and the white and black lines are showing different years of the turtle itself by looking at the different lags. Skeletochronology is usually done by looking at cross-sections of the humeri bone in sea turtles. Older bone will be in the center of the cross-section, as newer bone will be on the outside. But older bone actually gets reabsorbed. So how do we accurately find the age of the sea turtle? Looking at this image here, we have 6 lags that are already presented, and by zooming in further, we can look at the first annulus of the lags. By using this first annulus size and using a correction factor, we were able to estimate the age of the sea turtle. In this example here, this estimated age is around 10 years old. So what research actually led to this proposal? Kathleen Hunt has been doing Baleen Powder experiments since 2014. And on the right, you can see a picture of baleen being cut into. So on the bottom of it, there is the newest or the youngest baleen, and as you get towards the top, it gets older. So they're looking at different sections of this baleen and they're going to analyze it for different hormones. In this experiment itself, they looked at progesterone levels, and as you can see from this chart here, when there is higher concentrations of progesterone, they made this the pregnant years, and they are followed right after with non-pregnant years or very low levels of progesterone. And one cool thing that they found is the year following the high levels of progesterone, these individuals were seen with a calf swimming next to them. Kathleen Hunt's experiments are going to lead to our future plans for sea turtles. By using a cohort of sea turtles that were born in SeaWorld in 2007, and that were already analyzed by hormone analysis to show that they were already male, we have a both known age and known sex samples, which are going to allow us a proof of concept or to validate this experiment right off the bat. So the process itself is going to be taking these humerine cross sections. We're going to drill into these different lags, which are different years, and we're going to test the hormone levels. Specifically, we're going to analyze the testosterone concentration. By analyzing these different lags, we're going to be able to find an age of sexual maturity of these sea turtles. But why? Why is this important? For conservation management processes, we're going to be able to have a lifetime reproductive output on these turtles. We're going to find life stage models. We're going to be able to have population estimates. And we're going to have future management policies. These management policies could be protecting specific areas of the ocean that we know these turtles are mating, or areas that we know that they nest in. Of the 7 species of sea turtle, 6 of them are found in the United States, and all 6 of them are either endangered and threatened. So this process and this experiment can really help save these species for future generations. Here is a quick list of all the different studies that were referenced in this presentation. Before I end here, I'd really like to thank the Marine Turtle Biology and Assessment Program, or the MTBAP team. Especially in there, I would like to recognize Dr. Summer Martin for allowing me to have this position and giving me the opportunity to work with this great team. Dr. Cameron Allen has been my advisor throughout this entire year and a half, and she was able to meet with me on a regular basis to go over my project and help me out throughout the entire process of writing the paper in this presentation. Sean Murakawa is the Skeletochronology expert at PIFS right now, and it is really interesting to hear all about the entire process, and I'm glad that I was able to work with her throughout this time.